Right, so this is proteomics. New words for some of you. It all will be revealed, and this is from Canada.
Thank you very much. It's humbling to follow Dr. Bernstock. I'm honored to have contributed to one of your, thousands of books. Thank you very much. I was at the Department of Health and Human Services meeting a few years ago, and they wanted to change the name of chronic fatigue syndrome in the United States. And they had a terrible argument, so I got up and said, well, why don't you call it Baraniuk syndrome? That's my name. You can't see it from pronouncing it, but it's like the Three Musketeers. You get there in the bare neck of time. I'm learning that what I call chronic fatigue syndrome is a little bit different from what chronic fatigue syndrome is called over here. So coming from Alberta, this is the Carruthers criteria. We're going to call this the Canadian fatigue syndrome. My email addresses are there because I expect every one of you to call for cerebrospinal fluid, blood, urine, other samples that I just am tickled pink to be able to share with everybody. Up until, I guess, when I got here, this was the English view of fatigue, I take it. And of course, as my patients have taught me, many over 15 years, it's clear that this is an illness that's all in your head. Dan Claw, who was my division chief and worked very much in Gulf War and fibromyalgia, now interstitial cystitis, gave a talk one day, very dynamic, and then walking out of it, the cardiologists were talking about amongst themselves about the crazy loony ladies that were coming in, that it was all in their head, as if they hadn't learned anything from the damn lecture. And as you know, most of those people, classically trained, you're all pink elephants. This is Lucinda Bateman's figure. You've probably seen this many times of fatigue, the Venn diagram to chronic idiopathic fatigue, the overlap with fibromyalgia, the great sun that shines in rheumatology departments in the United States, the little brother, sister of chronic fatigue illnesses leading to the heart of things with ME. I have sort of found this to be difficult to buy dogma, so my tendency has been to sort of flip things upside down and take a fresh start at it. And one of the things is, if you look at the pain, as mentioned, we're talking about central sensitization. Now, it was very late when I made this slide. It says desensitization, but please forgive me. It's it's just ME, having done it. This accounts for the hyperalgesia, the allodynia, the pain and tenderness that we see so commonly. But many in the United States, chronic fatigue people and physicians ignore completely. You add the cognitive dysfunction. Has anybody with CFS, Canadian fatigue syndrome, got any cognitive dysfunction? We're looking at the fMRI of the brain defects in subgroups right now. What about autonomic dysfunction? Until recently, I thought this was kind of one of those things that, I was curious, but I didn't like to use tilt tables, so I didn't. But using other methods, I think that's very, very critical brainstem pathology. Irritable bowel syndrome. I'm seeing lots of Gulf War illness people because I have 2 Gulf War grants, and virtually every one of them have irritable bowel syndrome with diarrhea. to the point where I've got a poor guy who's got PTSD from his explosive diarrhea that he can't leave the house because he's afraid he's going to explode in the car. If his kids or his kids' friends are there, it'll be all over school, and that'll be it. So we got to help this guy. And recently, with all the lumbar punctures we found there were a lot of post-lumbar puncture headaches. And we were really, really concerned that we were causing spinal leaks and needing blood patches. But as it turned out, in talking to these people, they had extraordinary photosensitivity, phonosensitivity after the LP. And that was actually a trigger to cause them to have a migraine headache. Turns out 2/3 of migraine headache people meet the Fukuda criteria for CFS. In the Carruthers criteria, migraine is listed. And we've published now that 80% of our patients, both chronic fatigue and as yet unpublished in Gulf War, have migraines. So what's the link? Isn't migraine a legitimate disease? So it's all in your head, right? Well, I think this is what we would like to do to our Hippocratically trained physicians. And morph it further into it's all in your head. That, in fact, has become our laboratory mission statement. And I apologize to Picasso, Netter, and Harvey Ball, who invented the smiley face for our lab symbol here. But I want to also remind you that the brain may be where the problem is, but we don't know where in the brain to biopsy, and our IRB won't let us do it anyways. So we take cerebrospinal fluid. We have our most skilled people in the hospital do it, and it's a wonderful fluid for examining the effluent that comes from all of the neurons, the support cells. Very rich, as I'll show in proteins. So the hypothesis is it's all in your head. So central nervous system dysfunction is a critical component and related to the neural sensitization. Many neurotransmitter systems throughout the brain. With the cerebrospinal fluid analysis, we focus on the factors of relevance that we think will lead us to pathophysiological mechanisms of chronic fatigue syndrome. We want to look for diagnostic biomarkers for future testing, using cerebrospinal fluid first with the long-term hope that we'll find something that we can test in urine, saliva, or blood. And by identifying specific mechanisms, we can now target some of the ATP and other new mechanisms to try to improve specific components and ultimately the underlying problem in chronic fatigue syndrome. So this is the proteomics of cerebrospinal fluid. Now, proteomics, first off, is a study of all of the proteins at once. Oops. Judy, I found one of your mice. So proteomics is not that hard a concept. I mean, you've done ELISAs. That's proteomics, but it's one protein. You've done Western blots. That's proteomics, but it's maybe one, maybe a couple. But in this case, it's as many as your machine can detect. That's the only difference to remember. Now, what we do as a standard is we do a precipitation to try to clean up the proteins, separate the peptides from the big molecular weight, antibodies, albumin, things like that. Just about everybody will then chop these proteins up with trypsin into smaller peptides that can run through the liquid chromatography, the mass spectrometry steps, and ultimately be sequenced. Does anybody want to know about the quadrupole, time of flight, Orbitrap, ion trap mechanisms? Okay. Well, this is our wonderful first-generation machine, which we at one time could have sold on eBay, but had to pay somebody to carry away as a paperweight. It basically has a very high-powered HPLC here that takes nanoliters per minute. It's hooked into a first mass spec that will separate the different peptide ions, different other materials like metabolomic products. And then at the back, it has a special kind of time of flight mass spec that was probably worked on here in the Institute of ME because it's a very, very old and standardized method for measuring mass in electrically driven, magnetically driven systems. The great breakthroughs of the last 10 years, though, have come as well with the introduction of programs like Mascot and others that allow the bioinformatic analysis of more and more and more of the peptides that are found coming off of these machines. And as that has become a richer data set, we've needed to go to more and more sophisticated types of statistical methods. These, I won't go into at length, but they rival those used for microarrays and often are the same type. Now, our first study started in 1998. That was a year and a half after the word proteomics was invented. We took cerebrospinal fluid from about 62 fibromyalgia and Gulf War syndrome subjects. Turned out they all had chronic fatigue syndrome, but the people I worked with, they don't believe in CFS, they only believe in other things, so I was the only one that thought they had CFS. Fluid was analyzed by mass spec. We took 2 cohorts and pooled the secretions from 10 healthy controls, 10 illness subjects, and then we compared the 2 illness groups for those proteins that were present in the 2 illness groups and absent from the controls. Then after that, we looked at individual samples to see if the same proteins appeared. And much to my shock, we found a group of 10 proteins that were reproduced in the first set of 20 CFS people and the second set of 20 different ones. I'll go into what they do in just a moment. The odds of this happening were about 10 to the minus 15, so I thought it might be important. At the time, we classified these into protease anti-protease imbalance, which I think is a very important functional effect that we haven't started to look at really in CSF. We have innate immune and structural injury, and you'll see over and over again complement proteins appearing. Oxidant injury has long been described or ascribed as the mechanism of chronic fatigue syndrome, but the data is mostly in reviews from animals and not supported by human studies. Vascular regulation, we found these proteins which break down into about a dozen neurohormones each and have completely diverse, largely unknown effects. We thought we had leptomeningeal activation, but probably we're getting glial fiber proteins instead. And finally, we had a structural repair molecule here. So the thought was we have some kind of imbalance in proteases, activating immune system, oxidant injury, vascular dysregulation, eventually damage and repair. sounded nice at the time. Now, the machine we used back then could pick up a grand total of 84 ions. Yes. 15 peptides, limited numbers of proteins per runs. We had to use multiple runs. And then we moved with our collaborator who moved up to an Orbitrap mass spectrometer. He was able to go up to 7,000 ions per run, about 1,700 peptides and 170 proteins per run. This has since been improved. There are other systems out there, other laboratories that have much higher throughputs, but that's because we're counting our proteins in different ways. In the first of the Orbitrap studies, we compared the different databases that are out there for proteins. And there's one, for instance, the NCBI non-redundant NR. Turns out it has every single fragment of every single protein that's ever been entered into the NIH database. So it's a totally redundant, horrible, horrible database, but at least it lets you find everything. So it's one that we use. One of the things that it does is it comes up with lots of unknown or hypothetical proteins that turn out to be antibodies. And this has been a continuing problem. It's very useful to get rid of your antibodies here. You can see they account for a large proportion, about almost 2,000 out of our 4,000 total proteins that we found in this one particular study. Notice again a lot of complement hits, apolipoproteins, the protease inhibitors. Now, in this particular study, we also found that apolipoprotein E had an increased ratio and significant corrected T for an increase in CFS compared to healthy controls. And this seems to be continuing on, and I'm not sure about the significance of this yet. Clusterin, once again, is a regulatory protein in the complement system. We have albumin, but I think this is basically an artifact. We thought at first that this may be a marker of a change in the blood-brain barrier, so that you'd had more protein flooding into the cerebrospinal fluid in CFS compared to healthy controls. Serpin G1, serine protease inhibitor, also plays a role in complement activation. On the flip side, there weren't so many proteins that were higher in healthy controls compared to CFS. The glial cell protein I mentioned was one of them. We haven't reproduced any of these in subsequent studies. At this point in time, because the databases were so immature, we only really could pull out about 2% of the information. That is, out of every 100 ion peaks that we would detect after chromatography, we could only match about 2 of them to a protein. There's some other difficulties in that matching, but I won't go into it. One of the possibilities is that these other ion peaks represent post-translational modifications. For instance, are they oxidized? They won't show up in a standard screen. Are they other types of adducts? And our collaborators actually found some very interesting heavy metal adducts that are covalently bound to proteins in our set. That's not ready for press. The other would be sugars and especially lipopolysaccharides. The protease inhibitors, metal chelating, apolipoproteins were abundant, and once again, both classical and alternative complement pathway proteins. In general, these studies are are of the moderate to high abundance proteins, and they led us to now believe that the real meat is in the low, very low abundant proteins, probably derived from the brain. That leads us to Steve Schutzer's very nice series of papers. In 2010, they published a large study where they worked with the Pacific Northwest laboratories, which have one of the best proteomic facilities in North America. It's so good that Rudy Aberschold runs it, and he's also running the best proteomic facility in Europe, which is in Zurich. So it's got to be good. My first postdoc, or first PhD student was from Switzerland, so it has to be fabulous. They found 2,630 proteins in the cerebrospinal fluid. Previously, they'd found 3,600 in plasma. And curiously, about 56%, or about, say, 1,500 of these proteins appear to be absent from plasma. So they may well be unique brain proteins. Earlier this year, they scooped us, by coming up with a paper where they built on their earlier normal study by showing that there was a distinct proteome in chronic fatigue syndrome patients that was distinct from post-treatment Lyme disease. Now, this, I think, is very important because clinically these two groups can be difficult to differentiate. But the message from the paper is that when you look at mechanisms, when you look at protein biomarkers, it is possible to differentiate these two similar appearing illnesses. I would make the analogy to pneumonia. In 1600, you could do an autopsy on somebody with pneumonia and you would see pus filling the lungs. Right? So that's pneumonia. But after caulk, after staining, after sectioning, growth of bacteria, we now can understand that there's staphylococcal pneumonia, streptococcal, Haemophilus, etc. And I think we're moving in that type of direction. As we get the appropriate tools, we'll be able to sort through these issues much better. And just like in pneumonia, we'll be able to choose the best antibiotic, so to speak. Now, in these studies, I apologize, they didn't use English methodology. They took medically unexplained fatigue that met the Fukuda criteria. They excluded all psychiatric patients. So I don't know how applicable this is to over here. They had normal CSF and total protein counts, and they pooled all of these samples together in order to look at the total depth of the biomarker pool. And in addition, they ran 14 as individuals in order to compare between illnesses. For the Lyme patients, they had 25 who had been seropositive for Borrelia burgdorferi, had been treated for 3 weeks with IV ceftriaxone or cefotaxime, and had persistent fatigue and other neurological signs that had no explanation for at least 4 months. And again, they had individuals that they ran as comparisons. Now, one of the critical things in these studies is they took samples from very large, relatively large populations, and they needed at least 25 mLs to start with. That makes it it's not impossible, but it's a little impractical to use on a single individual at this point in time. They have to go and remove the most abundant 14 plasma-derived proteins. That means you remove about 95 to 99% of all of the protein. You're left with a tiny fraction. They then would do strong cation exchange chromatography. Don't worry about the details, except that that separates the proteins, and they took 30 different fractions there. So they were getting fewer and fewer proteins in each of their fractions. They did the standard trypsin digestion, reverse phase, MSMS, and an extremely complicated statistical analysis. They needed an enormous number of statisticians to work on this paper. If I go back to their paper, you can see that about a third of them are statisticians. That must be statistically important, right? Here was the breakdown of their proteins. In the normals, they had 2,600, in the CFS, 27, and in the Lyme patients, about 2,700. You can see in the normals only, there were 724 normal-only proteins, 734 only in CFS, and 692 only in Lyme. In their paper, their preliminary analysis is rather limited, and they point out that just an example of one protein, this CDK5, which is a signaling protein in Alzheimer's and Parkinson's disease. And they just raise the speculation as to what sort of neurodegeneration does this imply. They had 305 that were shared by these two, CFS and the Lyme, and from those they did statistical analysis and found 59 proteins that were different between these two subsets. When they did clustered hierarchical clustering, unsupervised structural hierarchical clustering, they were able to find that the proteins for the CFS group, these are the proteins listed here, the heat map indicates the degree of correlation, the significance of the correlation. And over on here, you can see the Lyme disease people, and all the red indicating high probability was in the Lyme disease people, and these, which would represent the opposite direction of significance, occurred in the CFS group. And furthermore, by cluster analysis, unless you're a statistician, this won't make much sense, but basically they were able to cluster these so that you could pass a piece of paper between the blue circles and the red circles, which is really, really exciting if you're a statistician. They took a few selected proteins from the set that was different between Lyme and CFS, and also proteins that were shared between the two. This is an example of proteins that are shared by CFS and Lyme, but were present in significantly higher levels in the Lyme cerebrospinal fluids. They're complement factors. Complement factor C1s, C1q, the B variant, and C1q, the C variant, and complement C4b. Now, I'll make a point here. It turns out that this is complement C4. The protein C4 has copied itself, duplicated itself. There's a couple of amino acids different, but you can't tell the difference between C4b and C4a except for about 3 amino acids. So when you see something like this, in my book, it should actually be C4A, comma B, because you can't tell the difference based on most of the sequence data. Now, as I said, it's mostly Lyme, then CFS, and much more than in healthy controls. Up here they have another heat map correlation showing that the ones in red were more present in the Lyme disease, and they're marked by the red proteins in each of these complexes. So that this would suggest that activation of one of the three pathways in complement leads to activation and plays a role in the Lyme illness. The inclusion with CFS would suggest that this innate immune system is also involved in chronic fatigue. They also did their receiver operator curves and found that C4b was their most selective marker in this system. Their example for the converse was those proteins that are involved in axonal guidance, or the growth of neurons. And here the proteins were found predominantly in healthy controls, then the Lyme, and finally the CFS group. These include, in the Lyme group, the epherin receptors here, and these are involved in the intracellular membrane, where you have axon attachment, and then at other places along the path. It's not the entire pathway being marked out, but it's a suggestion that this may be downregulated in these two illnesses. So to summarize, before we get to questions, proteomics using pooled samples of many subjects will attempt to detect as many proteins as is practicable, as many peptides as is practicable. Unfortunately, that's about $500,000 for a single run. So I think it will not come down in price like genome sequencing. And it requires an audience like you of statisticians to figure out what's going on. The cerebrospinal fluid pattern, though, can distinguish between subtypes of fatiguing illnesses, the Lyme versus CFS facuta. Now, once a single protein has been identified as a putative biomarker, it can then be confirmed by comparisons between individual samples from the two disease groups of interest, other similar illnesses, and then healthy controls in order to make sure that you have a significant difference, a CFS-specific marker that isn't also found in a substantial number of controls. And then, of course, you need orthogonal methods to validate the biomarker. That just means you have to go and do some other different type of test in order to show that this protein is present in a higher level in CFS compared to the others. One of my interests from the start is not the individual protein or the individual peptide, but is actually what's the combination that is important or is unique to a single illness, such as chronic fatigue. In our first study, we actually found a set of 5 proteins that had 80% concordance for diagnosing whether a person had CFS or was a healthy control. The signatures means that you may have 5 or 6 markers but that may be much, much more powerful than just a single marker test. The candidates that you identify in terms of regulatory signaling, inflammatory, and other mechanisms are going to also be very critical, because that will allow you to identify underlying mechanisms involved in the pathogenesis of the disease. And of course, if you can identify unique pathways, then that will lead to new drug development and novel therapies. We're probably 5 to 10 years away from seeing any fruits of this type of work. An additional thing is that a lot of the information, when you only look at the proteins, is lost in all of these other peaks that can't be sequenced. And we're working quite aggressively to try to find out if there are post-translational modifications like oxidation, nitrosylation, etc., that could implicate other mechanisms in the pathogenesis. And from that information, we hope to be able to come up with, again, new concepts of disease and treatment. So for the future, this method is going to allow us to define these illnesses based on the pathophysiological mechanisms. We may find that people change depending upon whether they have a flare or go back to their baseline. There may be differences depending upon severity of disease, if we ever figure out a way of assessing severity in a reproducible, agreed-upon way. It's going to take multidisciplinary outcomes and additional studies in order to identify those phenotypes. And I'm a strong believer that we will still need randomized placebo-controlled prospective studies in these well-defined phenotypes in order to prove that certain treatments are useful. I think just using open blind or single blind studies are going to potentially lead to large placebo effects where people could be placed potentially at risk to treatment, and they may, you may not know that until you do a placebo-controlled study. Last thing I wanted to mention is we're working on a Gulf War illness cohort. And Gulf War illness is a little bit unique. It shares many of the common features of chronic fatigue syndrome. But it was a 10-day war, or no, it was a 100-hour war with the air campaign before, and then blowing up Camisea, potentially spreading munitions over Kuwait and Saudi Arabia, plus having multiple sets of immunizations, some of which the Pentagon will not disclose, some giving gamma globulin, all these potential unique cohorts of people. But the bottom line is that 250,000 U.S. troops out of 750,000 came down with Gulf War illness. So if 1 out of 3 people in London got sick with this, do you think anybody would be interested?
Yeah, exactly. Absolutely right.
I think I'll just... So, in closing, I think we got to turn some of this stuff over on its head. As I mentioned at the NIH meeting in early April, I think we've got to have a Manhattan Project to move forward with new technologies, new concepts, with a better relationship and interaction between research groups. Because I think once we do that, we'll see that this set of illnesses is a completely different animal from the way it's being portrayed now. And then we can get on with some useful therapies. I'm pleased to share all of my samples with anybody who wants to do good research studies. You have grant money, I have samples.